receptors and learn how they fit into what we know about protein signaling to contextualize our paper discussion. Opioid receptors are G protein coupled receptors. They are located in the cell membrane of our neurons and stimulate a response with similar parts to the beta adrenergic receptor, but the parts are slightly different, as you'll see. Opioid receptors are involved in pain management, which is how many people took them and got addicted. We'll learn more about the structure of the mu opioid receptor in class. There are 4 classes of opioid receptors: the mu, which our paper is about, the kappa, the delta, and the nociceptin opioid receptor, or NOP. The NOP is structurally related, but is not generally considered in discussions of receptors, therefore we will neglect it from here on out. These 3 receptors are all involved in the body's response to drugs, but in the name of simplicity, we are only going to focus on the mu receptor today. To start, let's review how G protein-coupled receptor signaling works, as the mu opioid receptor is a G protein-coupled receptor. When the ligand binds to the receptor, it causes a conformational change in the G proteins, which then release a GDP and bind a GTP. Once the G proteins are activated, the G alpha dissociates, it wanders over, and it activates adenylyl cyclase, which turns ATP into cyclic AMP, or CAMP. The CAMP then activates protein kinase A, which phosphorylates a cascade of proteins that result in the physiological phenomena we associate with being scared out of our minds: a racing heart rate, breaking down glycogen for energy, and heightened senses. The players in the mu opioid receptor are similar, but there are some key differences. Instead of the GS alpha protein that we see interacting with the beta adrenergic receptor here, the mu opioid receptor prefers to interact with the G alpha I protein instead. The GAI protein actually deactivates adenylyl cyclase, which turns ATP into a cyclic AMP, and this leads to a decrease in the cyclic AMP concentration in your cytoplasm. One way to remember this is that opioids cause you to have the opposite response that adrenaline has on the beta adrenergic receptor. Your heart rate drops, which has a calming effect. Additionally, beta-arrestins also play a crucial role in opioid receptor signaling. It's true that when beta-ARK phosphorylates the receptor, it is tagged to be recycled by beta-arrestin. However, this phosphorylation can also trigger a completely different chain of events. One that may be responsible for the euphoric side effects associated with opioids. Here is a diagram of the cell's response to when an opioid agonist binds the opioid receptors. When the agonist binds, G-alpha I is recruited to the receptor. The G-alpha I activates a potassium channel, causing potassium to leave the cell, whereas the beta subunit inhibits the flux of the calcium receptor. Then, when the G-alpha I is inactivated, the receptor is phosphorylated and attracts the beta-ARK and the beta-arrestin proteins. At this point, the protein can be recycled, or it can trigger a different pathway that we haven't seen yet, and we're not going to go into a whole lot of detail on, the mitogen-activated protein kinase, or MAPK, signaling pathway. You can think of this as being an analogous pathway to PKA. It is believed that certain MAPK pathways are linked to the euphoric effects associated with opioid agonists, but we do not yet have a definitive picture. Now that we've established what opioid binding triggers in the cell, you may be wondering what causes opioid tolerance, or why addicted persons have to increase their dose to still achieve a high. The short answer is that we don't know, but there are some factors that may play a role. It's tempting to believe, and this was thought to be the case for a while, that the recycling of receptors from the surface would cause a resistance to the drug's effects. However, scientists now believe that recycling receptors actually has a protective effect against desensitization. Another factor has to deal with cyclic AMP concentrations. Although our cells are decidedly not at equilibrium, they like to stay at the steady state or keep the concentration of each molecule relatively constant over time, especially if that molecule is needed by other pathways. So if the opioid binds to the receptor and leads to a chronically depressed or decreased concentrations of cyclic AMP, the body will come up with coping mechanisms to compensate and increase the concentration of cyclic AMP back to normal levels. One of these mechanisms would be to make more adenylyl cyclase. Therefore, the person has to take more and more opioid to take the cyclic AMP levels back down to what they were before when the person first started taking the drug. Remember, cyclic AMP and pKa are key to the effects on respiration and heart rate. The third explanation, which we don't know a whole lot about yet, is that receptors may desensitize with time, or the downstream proteins may uncouple from the receptor. The phosphorylation of the cytoplasmic side of the opioid receptor is key. In fact, the receptor can be phosphorylated at 15 potential locations. The identity of the phosphorylated residues, the amount of phosphorylation, and the pattern are key in determining which pathways get activated. The way that MAPK signaling progresses depends on these phosphorylation patterns. It's possible the phosphorylation pattern of the receptor may be altered in an addicted person versus a non-addicted person and lead to different signaling. It has been shown, for instance, that different drugs, such as morphine versus fentanyl, lead to different phosphorylation patterns. While this may play a role in the desensitization of the signaling to the agonist binding the receptor, this, along with the effects of cyclic AMP and calcium and potassium ion channel regulation, likely play a role. We just don't quite have the complete picture yet. Opioid signaling is so complicated that different drugs lead to different responses in the cell. Just because a molecule binds doesn't mean the effect will be uniform. For example, morphine and fentanyl have different effects on cyclic AMP production, but similar effects on MAPK activation and signaling. Therefore, one of the potential ways to treat addiction is to develop drugs that satisfy the craving but don't get you high, although it's a lot more complicated than it sounds. Now, I'll walk you through the categories of molecules that bind to the mu opioid receptor. The first class are the ligands, or the native substrates for the receptor. The native substrates for opioid receptors are opioid peptides, including beta-endorphin, endomorphins, and enkephalins. These are released to receptors in response to something that is simply delightful, and you get a reward for it. Now we're going to move on to the classes of molecules that you can take with the intention of triggering the same responses of your native endomorphins. Agonists are molecules that bind to receptors and act like ligands, but they aren't actually the ligand. You've probably heard of many of these bad boys before. For the mu opioid receptor, agonists include oxycodone, morphine, heroin, codeine, and fentanyl, among others. You'll notice here that these drugs have fused fibrine structures, and if you look closely at morphine and heroin, the structural difference is the addition of two acetyl groups on heroin. Methadone, a molecule commonly used in opioid addiction treatments, is also an agonist, but it has very slow kinetics. This means that the effects of the molecule act so slowly, but if taken as intended, the person will satisfy a craving but without achieving a high. The next type of drug is the partial agonist. This molecule binds to the receptor and activates similar signaling, but some now has less receptor activation than agonists do. Ultimately, the effect of the partial agonist plateaus with dosage, which makes it difficult to get high. One example is buprenorphine, which satisfies cravings, but it's harder to overdose on. The principle of partial agonists is represented by this dose-response relationship plots. With agonists, the response increases with the dose of the drug, but with partial agonists, the effect eventually levels off with the dose, which would make it harder to overdose and would decrease the incentive to take more drug. The next class are mixed agonists, which have a mixture of both agonist and antagonist properties, which vary by dose or by receptor. Buprenorphine is also an example of a mixed agonist because it is the agonist for the mu receptor, but an antagonist for the kappa and delta receptors. Biased agonists are drugs that bind to the receptor and cause it to favor different pathways. This is a relatively new class of drugs that we're looking at. One recently discovered molecule is PZN21, which was found and hypothesized to cause the mu opioid receptor to favor interacting with G proteins over beta arrestin. Developing drugs that bind but downregulate the pathway that can lead to euphoria are a potential opioid therapy. The last class of molecules are antagonists, which bind to the receptor but inhibit any signaling. These molecules usually bind so tightly that they prevent other molecules from binding the receptor. Examples are naloxone or Narcan, which is administered to overdose patients. Naloxone binds so strongly that it outcompetes opioids that are already bound, making it an effective treatment for people with opioids already in their system. Another example is naltrexone, which is used as a therapy for opioid addiction and is particularly convenient because it needs to be administered only once per month. You may be wondering, if antagonists can just bind the receptor and prevent the high, why not go that route for therapy? It's one option, but therapy is much more complicated than that. The antagonists bind but deactivate the receptor, which means you'll still have withdrawal symptoms or dope sickness. This is a valid method of treatment, but patients need to be completely detoxed before they can begin. Once the patient is clean, they can take the drug to saturate the receptors, which makes it less tempting to take drugs, and if you do, you won't get high because your receptors are already bound to something else. Naltrexone is currently used, and its formal name is drug name is Revio or Vivitrol. Its monthly administration is a big advantage for busy people who need to go in and get treatment. However, it also has clear disadvantages. For one, it can be really hard for addicts to be clean since they have to go through dope sickness. So this is certainly a tougher method of treatment. Then why not just simply use a partial agonist? Well, if you use buprenorphine in the way it's not instructed to be, you can abuse it, and it does have value on the street. So partial agonists can be used, but there are certainly drawbacks. One solution is to use a combination of a partial agonist and an antagonist, specifically the mixture of buprenorphine and naltrexone in the drug treatment Suboxone. This way, patients can satisfy their cravings and simultaneously block a portion of their receptors at the same time, which makes it tough to get high. But it still allows the person to be a functional human. As you already have an understanding of, and you will become even more keenly aware of after reading the last couple chapters of Beth Macy's book, opioid addiction and treatment is a complicated and emotionally loaded issue. Macy talks about how the odds of recovery are much higher with medication-assisted therapy, just like the kinds we've discussed, but they're still not completely adopted or embraced by society. Why is that? Well, for one, it comes with a huge social stigma. One argument is that it's a really bad idea to treat one drug with another drug, and many people and recovery organizations will not help recovering addicts who take an approach other than complete abstinence. Stigma aside, there's a lot of practical issues associated with treatment. Once in the process of rehabilitation, many recovering addicts have a hard time finding steady work, and if they are working, it's often in minimum wage jobs. Treatment is very expensive, and is usually only available during business hours. For a patient working a minimum wage job 60 hours per week, it can be nearly impossible or awkward or difficult to ask for peak hours off when you're already being employed by people who, at least perhaps in their mind, are taking a chance on you. Then there's always the consideration that while these drugs are often the last lifeline for people in a desperate situation, they're still drugs, and many are sold and abused on the street. This is a public health crisis that will be up to my generation and your generation to solve. Hopefully, by understanding the biochemistry behind how these drugs and drug treatments work, we can develop better therapies